So chapter 15 is all about genes and how they work and this idea of transcription and translation. I do want to clarify this because down on your notes from chapter 14 it said that synthesis only occurs in the 3 to 5 direction, which it does. Okay, it does occur in the 3 to 5 direction. So if you follow the little bouncing ball, it occurs in the 3 to 5 direction. And I think actually it says synthesis occurs in the 5 to 3 direction. What that meant was the nucleotides were added in the 5 to 3. So the 5, remember, that's the ribose side, and this is the phosphate side. So there would be a phosphate, ribose, phosphate, ribose. This would be ribose, phosphate, ribose, phosphate. Okay. So the nucleotides are added in that 5 side first, but it's actually read in a 3 to 5 direction. Okay. Again, these are the Okazaki fragments, and just like how Emily had to do, she started right here, and that is a primer. And so she started down here, and she read for a while, and then ran out of room. And she had to go up further up where the helicases had already wound, and then read a little longer, and then run out of room. And then go up further where the replication fork was, and then read a little longer. And then John had to come along and ligate it all together.
So the Okazaki fragments, they're just floating around until they need to be used?
They're not necessarily floating around. The old strand, this dark blue strand right here, that has actually hydrogen bonded to the extra pieces of Okazaki fragment. Okay, so the Okazaki fragment is merely only one side of the sides of the ladder. Only one side of the rail is broken apart. So it looks something of this nature. So if you looked at a ladder, an Okazaki fragment ladder would look something like this. So here's your little rungs. Those are the hydrogen bonds between each, and it looks something like this. Like that. And so what the ligase does is it comes in and it blends these two pieces together. And it blends those two pieces together.
Oh.
Okay?
With a covalent bond?
It's covalently bonded because it actually adds that phosphate in and it bonds those pieces together. Okay? And again, remember that would then be the lagging strand itself. And the last part of chapter 14, I didn't really hit this last time just because it was all in the book, and it really got into telomeres and telomerase. But remember, telomeres is the end of DNA. Remember, DNA doesn't start replicating at the very, very end. It's going to get chopped off. Remember, it depends upon how long you live is how long those telomeres are, right? And remember, during embryonic development and during places like lymphocytes, which you need to have the full set of DNA, which allows you, remember, lymphocytes are those things that affect infection, as well as places that are being used frequently, like the inside of your stomach and the inside of your intestines. Those places need to live and replicate very frequently. So the telomerase, which is the enzyme that repairs those telomeres, are much more active a longer time on those cells, as well as in the embryonic cell, because again, you don't want your DNA, when you're developing, to start shortening all of a sudden before you're even 2 years old. So, and they've also found that, remember, and I don't have it up here, but remember this also does relate to cancer cells. They found that cancer cells, their telomerase is actually active. And so that's why cancer cells don't die off. They don't just naturally die off. Your cells naturally die off. But cancer cells don't naturally die off because the telomerase is active at that point. Just some kind of background information. Remember, this does all relate to Mendel's peas plants. Okay. Remember, Mendel was able to determine that if you had a father or if you had the male plant, and the male plant started with green peas, and green was recessive, and the female plant was yellow peas, the yellow would be dominant over the female. That gene that coded for green peas, that gene that coded for yellow peas, he didn't know anything about genes, he didn't know anything about chromosomes and that kind of stuff. But he understood there was some kind of significance there. So that then comes to what's called the one gene, one polypeptide hypothesis. Essentially what that says is that if every gene codes for only one polypeptide. Now that's a hypothesis, hasn't been fully proven yet to be a law or a theory, but they have found that to be the case for prokaryotes. Okay, bacteria, archaeobacteria, that is pretty true. One gene, one chunk of the DNA codes for one specific protein. So if you're going to make a different protein, it's going to be found on a different part of the genome. But in eukaryotes, it's not really been found that way. It's been found that it takes a little more than that one gene. That one gene might code for multiple different proteins. Or that gene plus another gene kind of merge together and code for that specific protein, whatever you need. So there is a one gene, one polypeptide that basically just says that that one gene codes for one specific protein. But in eukaryotes, it doesn't always hold true. You do need to know the difference between DNA and RNA. What are the three main differences here?
RNA has sugar ribose.
Okay, so RNA has sugar ribose, whereas DNA has deoxyribose. Okay, the shape. What about the shape? Okay. And
It's a single strand. And
Okay.
the difference between like T and U.
okay, so DNA, on what Chaz was saying, DNA is a double helix, whereas RNA is a single. Sometimes it is a helix. A lot of times it's just linear. And then the T and the U difference. Remember, DNA is A's and T's, G's and C's, and RNA is A's and U's, G's and C's. Okay. And also, I mean, really, where do you find DNA? If you're eukaryotic, in the nucleus. Whereas RNA, yeah, it kind of, and it transfers, right? Remember, there's multiple different types of RNA, and we'll go into this. You also need to know the transcription and translation. Remember, we talked about that, I showed it in the movie. Transcription is where you're actually going from DNA to RNA, and translation is RNA to proteins. There are 20 essential amino acids. Those are determined upon by codons. Codons are 3-letter nucleotides. Okay. Now there are more than those 20 amino acids. Do you have to memorize those? No. Even when I was in college, yeah, I had to memorize them. I'm trying to think back to the old AP test. I think the old AP test even didn't memorize. But you do need to understand the 20 amino acids, what they look like. I think in my animal science classes I might have had to memorize them, and biochem, when I got to junior level biochem, I did. I had to know how to draw and draw them out and stuff like that. All right, so here's that idea that I was talking about earlier, that, remember, you have DNA, and then a gene, and then DNA, and then a gene, and DNA. Okay? Now, inside these genes, there are specific introns. So remember, both prokaryotes and eukaryotes go through transcription, translation, and then proteins. Okay. Here's that codon chart. This is from your book. No, sorry. Yeah, your book has one, and it's all kinds of graphical and stuff like that. But you do need to know how to read these. Okay. So I kind of gave you a couple examples. We'll do some other activities with it later, but you do need to know how you do, first, second, and third letter. And remember, these are looking at the RNA itself. So if I give you the DNA, you need to be able to transcribe that DNA into RNA, and then translate by taking the RNA and finding the code. Okay. There's also a codon wheel, which I will give you as well. It looks just like this, but the wheel is actually a little easier. But this is the one y'all are trained for on the state test. Remember that? Yeah. But the wheel is actually much easier. It looks like this. Okay, so there's the wheel. I've never used the wheel, and when I didn't know about the wheel until I started teaching biology here. I really like it though, just because very easily you see what the first one is, A's, G's, U's, or C's, and then you say, okay, it's a U, and it's a UUC, so you say UUC. So very easily it tells you exactly what it is. So this one, to me, actually works much quicker. They will give you this chart though on the AP test. So be more comfortable with this one. But once you get comfortable with this one's fine too. I'll actually give you both on the test and here in class. Yes. Yeah. Yeah. And all right. So prokaryotes. Remember, prokaryotes do not have a nucleus. Okay. So the reactions are coupled at the same time. At the same time, where mRNA is being formed, okay, ribosomes are attaching to the mRNA. And from those ribosomes, new polypeptides are being formed. So it's all at one step. It's not like you have to go, and it's not like when you go through the drive-through, you're like, okay, pull up to the first window, right? And you put your order in, and so pull up the first window, you pay your money, and then you go up to the next window and you get your food. It's all in one place. You pay your money, you get your food. You take your order, you pay your money, you get your food, all in one window. Okay? Whereas in eukaryotes, it has to do that format. Because in eukaryotes, remember that the mRNA has to leave the nucleus to go to the ribosomes. So they're what's known as a coupled reaction. It's occurring at the same time.
Transcription and translation evidence.
Yep. Because again, like these little yellow dots right here, those all represent ribosomes. So you notice that the ribosomes are attached at multiple places along the mRNA, and so every single ribosome will then code for a different type of amino acid, or a different type of polypeptide. Now, here's the steps of transcription. The RNA polymerase will add at the 3' end. Now, there are specific sections. This right here is what's called the promoter section, right there, that yellow section. Okay, that promoter section is what starts it. Now, where does our initiators come in? Well, our initiator is actually way down here. Okay, and our initiator comes up here and says, "Hey, here's that TATA box." Okay, and it joins all the transcription factors. The RNA polymerase then comes up behind the helicase and finds that promoter and then continues on. Okay. And we'll do a simulator next time in class. Actually, we'll be in the computer lab, and I'll show you this really well. So that's where the RNA comes off, and then finally it terminates. So again, this does move at 60 nucleotides per second, and it starts at the promoter, and it ends at what's called the termination site. Remember, RNA does need to be processed. Kind of went through that. This picture over here is kind of going through the idea of how these introns are going off. These are called snRNAs. I would just remember SN as in SNP. What they do is actually branch, and they snip the intron out. It's called a lariat. And then they merge these two exons together. And what was the benefit of this poly A tail? Why would you want a bunch of A's at the very end?
For protection.
Yeah, for protection. What's the benefit of the G prime cap? Same thing, right? This is kind of like a front end bumper, right? You hit a tree, not a big deal because you got a bumper. This is like your friend that you take in the woods with you, just in case there's a bear that's slower than you, right? Because you don't really have to run faster than the bear; you just have to run faster than your friend. Okay, that's what the poly A tail is. So if you're the one that gets invited to the woods all the time, I would not join track. All right, so this is tRNA. This is the structure of tRNA. It does form this kind of clover leaf structure. The very end of it is called the anticodon loop. This tRNA is what carries the amino acids. The tRNA, again, has an anticodon. The structure does fit its function. So remember, it has a specific place for the anticodons. It has a specific fit that it will fit into these ribosomes. And there's something called what's called the wobble. No, not that wobble. What it means is that if you don't have exactly the right anticodon, but it needs to have that amino acid right there to form the right protein, it can actually wobble itself into the right position. Okay, so say you need like an AGU, and you have an AGC. Close enough, right? And it can actually move itself into there. It can wobble itself into there, and it can find the correct format. Okay, so it knows its code, and it knows that this amino acid needs to be connected to that amino acid. And if you notice that there's 20 amino acids, right? But there's way more possible codons. So not every codon codes for every single amino acid. Codons will code for more than one amino acid. Okay, because if you go back to this wheel here, right? If you look, all these are all arginine. So CGU, CGC, CGA, CGG will all code for arginine. So even if it should say CGU but it says CGC, it's okay. And that's that wobble idea. Right? Now there are 3 different sites, the P, the A, and the E. The P site holds the growing polypeptide. That's where as the amino acid is coming in, as the tRNA is coming in, the amino acid is joining together on the P site. The A site is what holds the tRNA with the next amino acid. So it's kind of like a little sequence. The P site's there. It's got the polypeptide being formed. The P site's right there. The A site's right there. And so the A site moves into the P site, And so the new that tRNA moves out of the way. The new tRNA comes in and joins its new amino acid to that polypeptide. And then another tRNA comes right here onto the A side. And then the ribosome moves down a little further, and that tRNA that was there now kicks out, and the new amino acid joins to the existing polypeptide. So it's a continual sequence. It doesn't have to use energy, and peptide bonds are formed. That's why it's called a polypeptide, because peptide bonds are formed between each amino acid. There is a final touch to those proteins. There's a final, just like how the mRNA has to be processed, it's the same thing on the proteins themselves. They have to be correctly folded and bent, like how we talked about before on the macromolecules, where they go into the chaperone and they bend and they form the correct way to form the secondary and tertiary structures. Okay. Here's the elongation sequence. I put that really low. So this is what this is talking about. So if you look at it, so there's that P site, there's the A site. So let's just start. Okay, so it says right here, it says matching tRNA and a codon with mRNA codon, which is right here. So a new one's coming along. Okay, so here's the new tRNA. There's the anticodon. The anticodon is going to match up with this codon. What is this yellow right here? What's that yellow? If that's tRNA yellow, then what's this long, skinny right here? It's RNA, right? Which kind of RNA is it? This is mRNA. And so what's this big globular orange thing? This is outside of it. This is the ribosome. Because what's going on here? What are they creating? A protein. And so ribosomes, its job is to create proteins. So this is what's going on in that factory. So the P is holding on to the growing polypeptide. The tRNA comes along, moves on to the A. The A kicks over, the P kicks over the A, and then the new polypeptide joins. So you see how there's two here, there's one there, there's three. That P then goes away. It goes into the exit site. Okay, so this one, so again, distract this one. So this guy stays right there, stays right here. It says, okay, now you've got A here. You're going to use GTP, which is the same as ATP, just instead of adenosine triphosphate, it's guanine triphosphate. This polypeptide moves over to the A site, so now we have three. And as the RNA is being fed, it's going to kick the empty tRNA out of the E site, and now you're ready to go for a new tRNA. And it does that over and over and over again.
What happens to RNA after it's done with the ribosome?
So what happens to the RNA? So the tRNA will go back and find another amino acid. So the anticodon, it knows that AUG is a methionine. And so it goes and goes into the cell and finds another methionine amino acid. And it's ready to go for the next AUG that comes up. The mRNA, after it's being read, it is broken down into its parts, into the different nucleotides, and then it's being reused. So the nucleotides, all y'all that were standing out there on that side, you would then, after you were formed into an RNA, you were then read, translated, you would then be broken apart again, and then you'd be reformed later on. Next time an RNA polymerase needed you, or next time a DNA polymerase needed you. The tRNA doesn't really ever the tRNA goes and finds the amino acid, and then our RNA, this orange globular thing right here, this ribosome, this is actually our RNA altogether. So that's actually a big chunk of RNA inside that makes up the ribosome. Now, there are some mutations that occur, and you need to look at the different types of mutations in the book. And I didn't really put it here because I think I didn't want to hit it too hard here, but just know that there's a difference between like a deletion mutation and a friendship. It's kind of like this.
Do we turn that packet in?
Yes, I'm going to pick it up.
What about the
Okay. So fat cat sat, right? So this is what a mutation basically says: is if you have an addition mutation and you put an S right here, okay, and you're reading this, now all of a sudden this is a codon. This now becomes a codon, and this now becomes a codon. Okay? So all of a sudden it shifts that frame to the right one. That's if the S is there. But let's say instead of adding S, we take away the C. Okay, so this is a codon, this is a codon, and then that would end up being a codon as well.
So what if you have two mutations?
Okay.
Bump it back?
It could bump it back.
I feel like lost one.
It could, yeah. There's also something like this, where let's say that, yeah, you do the frame shift mutation, but it doesn't actually shift it. Okay? They go through and they add another letter there, but it adds it to where now the code is still read exactly the same way. It was what's called just a nonsense mutation. So it doesn't even change the actual function of it. You could also do something like this where you have a complete addition of that codon. Okay. What disease have we talked about that has a repetition? It's actually a dominant disease. There's a repetition of the same nucleotides. It made you, and determined by how many times that repeated. What? Yes. Huntington's disease. Depending on how many times that repeated, how many times it repeated cat, is how long you were going to live if you have Huntington's disease. Okay? Huntington's disease. Hunning.
Live for not a long time?
No, it was actually the more cats you had. Well, not the crazy cat lady, but the more repeats you had, the longer you would live. So let's say you had 10 repeats, you lived to 27, but if you only had 8 repeats, you lived to 25. It was like that exact. Yeah. So.
We try that away.